Awesome. All right. So how might a shrimp and a surgeon see eye to eye? It's the purpose of my talk. Brief introduction. My name is Iezzi. I'm a first-year PhD student in the Shtein lab in material science engineering, and I work with optoelectronics. So how does light interact with electronics? So what is the shrimp I'm going to tell you about? This is the mantis shrimp. It's super cool. It hangs out at the bottom of the ocean, and it's gotten a lot of attention in the press over the last few years, mostly for its punch. It actually can punch through a piece of glass about an inch thick. The big ones can, and which is really interesting, but if I was looking at robotics, I might be interested in that. But what I'm really interested in is actually its eyes. It's kind of crazy, but their eyes have really interesting characteristics that we're using to work on our work in our lab. So what if I told you that its eye could help a surgeon when he's inside someone looking at a tumor that he's trying to remove? Instead of removing a whole bunch of tissue that might be healthy, he might be able to only remove the part that is unhealthy right in real time. Or if you're at an assembly line where a bunch of food is passing through and you're trying to figure out, is this apple good or is this apple bad? Or is this apple going to be bad the next day or 2 days or 3 days in the future? How can we differentiate between those? Or even before you even get to that assembly line, like if you're out in the field and you're looking at, you have aerial imagery of a crop that says, okay, this plant is going to get sick within a week and we need to put fungicide or pesticide down because it's responding to a certain thing. So the mantis shrimp eye is something that we can use to be able to potentially get to those capabilities. But first I want to talk about like how do we actually see things? How do humans see things? How do other animals see things? So we all have what are called rods and cones in our eyes. The rods pick up low light. So if you're walking around at night and you can't really see too much, you have many of those to pick up the low amounts of light. But during the day, you use your cones. And you have 3 types of cones that pretty much absorb light in 3 different areas of the spectrum, which is red, green, and blue. And it's really cool because that's essentially how we've evolved and how many animals have evolved is we used what's available. So the solar spectrum, the sun, gives off light in the filled in curve on the back. And so you have a lot of red, green, and blue light. So we've developed capabilities biologically to be able to look at this light. But there's some limitations to it. So we have really good brains, and they can mix light very well. So when we see yellow or we see colors in between the red, blue, and green, that's because our brains are actually mixing those colors together real time. And that's how our cameras work as well. So you have a filter in front of the actual sensor on the camera that brings in red, blue, and green light and mixes them together real time to create that image that you just took on your camera phone. But the problem with that is that we all see colors differently. So I don't know if you guys remember this thing from a few years ago, but like people were getting all up in arms about like, is this dress black and blue or is it white and gold? And it really is, it's up to a lot of different things that I'm not going to get into right now, but a lot of it is because we actually perceive colors differently because your cones might be better than my cones, or my brain might not be quite as good as yours as mixing all the colors together. And so it leads to potential areas where there's too much subjectivity instead of objectivity. So why the mantis shrimp? It's really cool because it has 16 cones, and it actually can see light, the actual individual wavelength of light at 16 different areas of the spectrum. And so what that allows it to see, you actually can see, so where we see blue, the mantis shrimp can actually see 3 different blues that we would typically be mixing together. And so if you're looking at some blueberries, the mantis shrimp says like, oh, this blueberry is ready to harvest and these 2 aren't quite ready to go. And so that's really awesome. Like how could, like how could we, but how do we emulate that? How do we actually make a system that can do that? And it's called hyperspectral imaging, which sounds like out of like Back to the Future, but it really just means, I've been talking this whole time about there's like 3 bands. Hyperspectral just means you split up those bands into many smaller bands so that you actually can start getting signatures at individual wavelengths. And it's similar to how a lot of chemicals are analyzed today as you run through the system and then it has a bunch of peaks. And it says, okay, if this chemical will have a peak here and here and here, and then this chemical, which is completely different, will have a peak over here. And what it does is it uses the visible spectrum, what we use to see, to be able to create those peaks. So you can see like here on the, I guess the z-axis of this hyperspectral cube, you have little peaks. And so at this area on the actual image, you have this material or something like that. So one way to do it is actually very similar to how a camera works. So like usually you have a red, green, and blue filter on the front of the camera. Instead, you just have many, many more filters, and they're very, very small. They're actually about the size of the actual pixel of the camera, which are about the 2 or 3 times the width of a human hair. So very tiny, and you actually put these filters directly onto the sensor. But the problem is that these, right now how these are made is very expensive, and it's a materials limitation. That's why we're approaching it from a materials standpoint. One of these, which is about this big, costs about $8,000 right now. And it's the cutting edge, and they work really great, but $8,000, like if you had to go out in the field or something like that, that's a little bit too expensive right now. So how do we approach it? So how your actual cones work, they have what's called pigments. It's a natural color, and that's the dyes in your shirt. A lot of the color you see around you is from pigments. It's actually the material itself absorbing and emitting certain wavelengths of light, reflects some and absorbs others. But what's really cool is there's actually ways to create light without actually having a pigment. It's called structural color. And so if you have structures that are on the scale of what the wavelength of light is, you actually be able to kind of mess around with what the colors you're actually seeing are. And so you actually see the wing of a butterfly, it doesn't have any pigment in it. It's all structural color, but you guys all seen beautiful butterflies flying around. That's because of the structure behind it. So how do we do it? You actually take 2 transparent materials, ones that you could see through on any given day, and you make them super, super thin. And if you put enough of these materials on top of each other with differing refractive index, so refractive index, throw the pencil in the water, you see that it bends. 2 different materials like that. Throw enough of them on top of each other, you actually can allow only certain wavelengths of light through. And if you put enough of them together, you can actually allow, like what I was talking about, those very, very narrow peaks only through. So typically you use like hard materials to do this, but what we're actually using is the stuff you guys, if you haven't been in the nail salon, yeah, exactly. The acrylic, that's really similar to like the acrylic they use in the nail salon. It's called a photopolymer. You essentially use light to harden the polymer. And so what I do personally is I do simulations. So I say like, okay, given this thickness of material, given these mix of materials, what does the interface look like? What peaks could we expect? So like, okay, here, nice, you have a nice, really sharp peak here. Like, how sharp can we get it? And then we print with this really cool thing called an Iezzi printer. It essentially like uses electric fields to draw out droplets of polymer, and then we cure it immediately. And so on the bottom right, you actually see this is about 200 microns. So a width of a human hair is about 200 microns. So this is super tiny. And we can print these pixels all next to each other. And then the next step on the journey is to characterize it. So we use like a cool microscope that essentially looks at the reflectance or transmission of light through the samples. So that's a lot, but bringing it back together, what we're trying to do is say like, okay, how can you use the butterfly's wing, like the structural color of the butterfly's wing, to emulate a mantis shrimp's eye to be able to do something where you could get, for example, like real-time imaging, going to the doctor and they say like, okay, hey, you might have a rash that's going to start popping up in the next few days or something like that, to essentially give you more insight or be able to differentiate materials. So what does this mean to y'all? There's actually a lot of cool stuff out there online and cool demos that you can do with optics and imaging. So the thing on the left actually is it's a free online calculator where you actually could do very similar simulations to what I do. It's called Filmetrics. It's a reflectance calculator, and they actually have a huge database of materials as well. So you could just let them run with like the thicknesses and the different materials, mix them together, see what they like, who can make the best peak. And then if you want to do like real and the experimental stuff, this is a really easy one. You just like take a small glass, put it in a bigger glass. You have sunflower oil in there, and if you pour sunflower oil around it, the 2 indexes are the same, so your eye perceives it as the same thing. So the actual small glass disappears. It's pretty simple, but pretty cool. And then the inspiration for this talk came from actually a radio lab. from a podcast on Radiolab. If you guys haven't listened to Radiolab before, it's a really cool podcast that WMYC does. And they did it on the mantis shrimp. So with that, I want to say thank you, and if you guys have any questions, I'd be more happy to answer.